So I'll start with the first chapter in Unit 5. So this part was just an overview. That's pretty self-explanatory. It's interesting, though, if you try to look at the geology of Earth, how this really shaped it. The early Earth had so much CO2, and it wasn't until plants came along and started making oxygen, changed the acidity of oceans and everything else, the temperature of the Earth, everything. So these are just some basic structures and what some of the players involved do. So here's how NADH absorbs a hydrogen in 2 electrons, right? So here is NADH or NAD, NADH. If you put a phosphate group on it, then that's more involved with biosynthesis type of things. If it doesn't have the phosphate, it's more involved in getting energy. In other words, catabolism rather than biosynthesis. So flavin does the same thing, or similar thing. Here's the structure for coenzyme A. So you can see that adenine and ribose and phosphate and many of these things are just repurposed all over the place. Coenzyme Q, the lipid-soluble redox agent. So then we talked about mitochondria and chloroplasts and the different ways to make ATP. So substrate level phosphorylation, we saw that in a couple different places in the metabolic pathways that we looked at. And substrate level is meaning that it doesn't happen in the mitochondria, or I shouldn't say that. I said it doesn't require oxygen to do that step because, it's not requiring electron transport to do that. An oxidative phosphorylation occurs in the mitochondria. So that's basically 2 huge steps. You can break it up into individual things. So NADH reduces the electron transport system at complex 1. FADH2 reduces the electron transport system at complex 2. When you do that, you only make 2 ATPs. And so then we have the cytochromes, and then the chemiosmotic hypothesis. So electrochemical potential is created by the electron transport system. So you're pumping protons out as those electrons that came from NADH and FADH2 are reducing the electron transport system. So when you take a positively charged entity and you pump it outside, now you have an electrical gradient. And then it's also a chemical gradient, because if this is neutral and you just start pumping protons out, then this starts to become basic. So the pH is quite different between the matrix and outside the matrix, which is the intermembrane space essentially. That equilibrates with the rest of the cytoplasm. So now you have this electrical chemical gradient set up, and how do you turn that into ATP? Well, the protons go down the concentration gradient through complex 5, which is the ATP synthetase. And that's where conformational changes in this huge protein squeezes together ADP and phosphate, squeeze out of water, and you get ATP made. If you look at the photophosphorylation that goes in chloroplasts, it's pretty much the same situation. You create an electrochemical gradient, but that's driven by light. It's the same process. You pump protons, electron transport system pumps protons on the other side of the membrane, and you create an electrochemical gradient, and you make ATP when that electrochemical gradient is relaxed, when hydrogens go through the ATP synthase protein. So here's an overview of metabolism. So here's what happens to dietary proteins, dietary carbohydrates, dietary lipids. Those are the three major things that give us any energy whatsoever. So this is the catabolic pathways, right? As food comes in. And then we talked about, well, if you eat a lipid, can you turn that into muscle? Okay, so you go through this. Here's beta oxidation right here, and you get to acetyl coenzyme A. Can acetyl CoA go that way? No. It can go that way, but it doesn't do us anything. So to get it into protein, for example, the carbons have to go into the citric acid cycle. Some of the citric acid cycle intermediates can be taken out, in other words, shunted away, and turned into a biosynthetic pathway to make new amino acids. So that's at the level of citric acid cycle intermediates. But what about if you eat carbohydrates? Can that same carbon end up in a fat? Well, at the level of acetyl coenzyme A, you can go through lipogenesis, which if you remember occurs in the cytoplasm, and wind up in adipose tissue. So this figure is trying to show you the compartmentalization. So here's blood sugar. When it does come into the membrane, that's because there's been a process set up that puts the glucose transport protein onto the membrane, if you recall that. So most of the time that involves insulin. So the first step, glucose comes in, and the glycolytic enzymes are right there in the cytoplasm. And the first step of glycolysis is you create glucose 6-phosphate. That's cellular sugar. It can't simply go out again anymore. So blood sugar is glucose, cellular sugar is glucose 6-phosphate. So then that occurs in the cytoplasm. Then we saw the mechanism how to get pyruvate into the matrix, where it's then converted into acetyl coenzyme A. You make some energy here, and then you go in the citric acid cycle, and the whole purpose there is to make energy. So the two carbons that come in as acetyl coenzyme A are given off. And then with all the other processes, electron transport chain is where you make water. So the hydrogens that came in over here end up in a water molecule. That oxygen came from the air that you breathe. So we see also the beta oxidation of fatty acids. Conveniently, that happens in the matrix of the mitochondria because the citric acid cycle is right there. Fatty acids that may be carried around in the blood with lipoproteins, slowly broken down into fatty acids, they can be transported across the membrane. Now, what is it you're going to do with these? You could use these to synthesize triglycerides. You could use these for energy. And in one of the chapters, we talked about how that was regulated. The fatty acid synthesis occurs in the cytoplasm. And of course, it requires NADPH to reduce it. So biosynthesis are reductive processes, whereas catabolism is oxidative processes. So in general, you could make a big categorization and saying, well, oxidative, you oxidate with NAD. In reductive processes, in biosynthesis, you reduce with NADPH. So here's 3 ways to deaminate amino acids. So here is glutamic acid. Maybe I should. Anyway, so the first thing is you take an amino acid, pretty much any amino acid, and you want to take off its amino group. So it's done by aminotransferase, where you transfer the amino group from one to the other. So alpha ketoglutarate, which is not an amino acid, is simply an acid. It gets transaminated, and now you have glutamic acid. This alpha keto acid can go into the Krebs cycle and do all kinds of different things. And then if you look at oxidative deamination, this is just of glutamic acid. So glutamic acid accepts most of the amino groups, and then it's oxidized into alpha ketoglutarate. Well, alpha ketoglutarate you may see is in the Krebs cycle. And then 2 amino acids are directly deaminated by specific enzymes. So they go through a separate process than most of them over here. Why that is, I'm sure there's logic to it. I don't know what that is. So anyway, whether it's serine or threonine depends upon that methyl group right there. And then this goes off as ammonia. So in the cell, there's a lot of places where ammonia is being generated. Ammonia is extremely toxic. So for a terrestrial organism, such as ourselves, we don't have a way to get rid of it because we have to retain water so much. So we have to store it in a more safe manner so we convert it into urea. So here's the overall view of the urea cycle. And it looks like we're using a lot of ATP to do this, but that's not exactly the case. It's a very efficient process. So this is in the mitochondrial matrix. So when when it comes into the mitochondrial matrix, right where CO2 is being produced, it's almost instantly phosphorylated into carbamoyl phosphate, a high energy intermediate. It's found only in the matrix. So it combines with ornithine, another amino acid. Ornithine is found in the matrix and the cytoplasm, as with citrulline. So it forms citrulline, and then aspartic acid is needed. And obviously, if you ran out of aspartic acid, that would be a problem. So then you're using more ATP to convert this into this large amino acid called argininosuccinate. At the same time, fumarate is released in this next enzymatic step. The fumarate goes into the citric acid cycle and yields 3 more ATPs. So that means that you're not spending more than maybe 1 ATP to make a molecule of urea.
I have a question about the number 2, oxidative deamination of glutamic acid. If glutamate is like the primary neurotransmitter, why would we want to break that down? Just for energy?
Well, you don't want to have every glutamic acid used for that purpose, right? So glutamic acid is used in proteins. It's used in this process over here. It's used in a lot of different processes. It's just repurposed. So
okay,
what metabolic pathway is going on at that particular time in that particular cell is all regulated by the different points of regulation. So this is going on potentially not even in a nerve cell, right?
I see. Okay, thanks.
If you look at higher organisms like us, or I should say, you know, most of us, our brain is so separated and so treated so specially from the rest of our body. It's protected from processes like having urea build up and stuff like that. It's the blood-brain barrier and all kinds of other things. So I think your question really refers to, you know, the separation of functions and metabolic pathways by compartmentalization within the body. So anyway, if you look at arginine, that was the one that had that weird functional group, right? So you got something that weird, it doesn't even seem unusual that urea would come off of that. So let's see. I think I said most of that. So then glycolysis, and the purpose of glycolysis is to make ATP. And so here's the overall reaction. This is in the absence of oxygen, right? So you make NADH, but you can't oxidize NADH and make energy from it unless you take that to the mitochondria. Then it requires energy to do that. I mean, oxygen to do that. So the good thing about glycolysis is that you can still make ATP even in the absence of oxygen. The problem is you have to have enough NAD as well as this stuff. But NAD is really important. So if you go through anaerobic glycolysis and you end up with all your NADH tied up as, NAD as NADH, and now you cannot even make energy with that. So we looked at several processes to regenerate NAD so that anaerobic glycolysis can keep going on. So remember we had some setup reactions, right? So the purpose of those setup reactions was simply to get a 6-carbon molecule that is nearly symmetrical. So then when we cut it into 2 3-carbon pieces, those pieces can easily be interchanged back and forth. That's why in some books you'll actually see 2 glyceraldehyde 3-phosphates. They don't even talk about this necessarily. So from here on out, everything should be multiplied by 2. So we spent 2 ATPs. We haven't made any energy yet, but in the first step, I would make 2 NADHs, right? Because I got 2 of these. Then here I make 2 more. Let me go through some setup reactions, basically to end up with a high energy molecule called PEP, which then you can do another phosphorylation. And there's 2 of these, so you get 2 ATPs from that process. So this one right here is substrate level phosphorylation. So if you look at the control points, we talked about this simply because it's important, I guess, for is the cell going to keep that glucose or is it going to let it go to the next cell? So here's an enzyme that is an allosteric enzyme. It's activated by these, inactivated by those. Here's another one down here. It's activated by fructose 2,6-phosphate as well, inactivated by other things. So let's see. So here's how much total energy you can make. In heart and liver, it's more efficient because it's easier to get the NADH from the cytoplasm into the matrix. That transporter is more efficient. The skeletal muscle, less efficient. But without oxygen, you can still make 2 ATPs. Oh, that's it already. I was thinking of that as a long chapter, but I guess it's not. So any questions on this one? Okay, let's go to the next chapter. It's not what I wanted to open. Damn, this thing's, the windows cover everything up. In chapter 37, we look at some other pathways. So what happens to pyruvate, which is the end product of glycolysis? Well, most of it's going to go right into acetyl-CoA. And I'm emphasizing the fact here that you're making ATP. And this is basically in the matrix of the mitochondria. So they don't consider it part of glycolysis, and they don't consider it part of the citric acid cycle. And then pyruvate can be turned into lactate by animals, which ends up through the Cori cycle going back to form blood glucose, essentially. So you're taking pyruvate and you're reducing it into lactate. And the only purpose for doing that is to generate NAD to be used in glycolysis. So you can go back and make 2 ATPs from anaerobic glycolysis. If you're a microorganism, and I'm certainly not saying that you are, then there's a different process. Pyruvate is decarboxylated to acetaldehyde. Sometimes it's oxidized again into formaldehyde. And then you make NAD by turning that into an alcohol. So if you reduce aldehyde, you get a primary alcohol, right? So the only reason they do that is to regenerate their NAD for anaerobic glycolysis. So yeasts make ethanol. Most bacteria make methanol, which is quite toxic. But these two things are toxic to themselves as well. The only reason why they're doing it is because they need to regenerate NAD. This is fairly toxic to us. I mean, it can damage muscle tissue especially, but we're forced to make it. Okay, the Cori cycle, pretty obvious. The gluconeogenesis is probably easier to talk about it in comparison to glycolysis because it also happens in the cytoplasm in the same place. So here's glycolysis in the red, and here's gluconeogenesis in the blue. So at the same time, I tried to show a bunch of control points. The purpose of control points would be, this is in the same compartment in the cell. It doesn't make sense to be burning glucose for energy at the same time you're taking pyruvate and turning it back into glucose. Makes no sense at all. So you notice that things that activate these enzymes will inactivate these enzymes. And I don't need to go through every single one because it's just written right there. And then if you look at where there's control points, those are different enzymes, right? Why is this the same enzyme? Because the same enzyme will catalyze both directions for a reaction, right? Here's another control point where you have two different enzymes that are turned on and turned off by opposite things. And then this is not regulatory, so the same enzyme, same enzyme, same enzyme, same enzyme, same enzyme, same enzyme. And then you get to a control point, they have to be different. And like we were saying, PEP is a highly energetic molecule. So it's so energetic that you can make an ATP, substrate level phosphorylation, in this step right here, because it's such a large drop in energy, potential energy. But what that means is, like, if this is a long way down to the next step on a staircase, how do you get back up to that step on the staircase? You're going to have another step. So this is broken up into two steps, and one of them is carefully controlled. So we see another point where oxaloacetate is used. I've always meant to do that, make like a little box or something talking about the role of oxaloacetate. Maybe I will, I'm writing it down, doesn't mean I'll be able to get to it. Okay. So glycerol, what happens to glycerol? It can be turned into dihydroxyacetone phosphate and essentially enter the Krebs glycolysis. So here I was trying to make you think about this. If the cell has high energy, that means it's probably not going to need more energy. What are you going to do with all that energy? Well, you store fat, store glycogen. In other words, you're biosynthesizing. So these are the things that would indicate a high amount of cellular energy. These are the kind of things that would indicate low cellular energy. So in that situation, you would turn off biosynthetic processes and turn on catabolic processes that are designed just to make energy. So the role of fructose 2,6-diphosphate is rather complicated, but there's an enzyme complex, which I drew right here. It has two enzyme activities on it, depending upon the state of phosphorylation. So this is how insulin affects it, and glucagon. So let's see, I looked at glucagon first. So glucagon, through cyclic AMP as a second messenger, turns on protein kinase A, which is a phosphotransferase. And what does it do? It puts phosphate on proteins. So this protein is just sitting there, if this is in particular a liver cell. And when you phosphorylate this protein, you inhibit one activity and you activate the other activity. So when you do that, you have less fructose 2,6-diphosphate that turns off glycolysis, turns on gluconeogenesis. It increases blood glucose levels. That's really what it does. What about insulin? Insulin binds to its receptors. That receptor itself is a kinase, so it turns on protein kinase B. And so what does that do is it turns on a phosphorylase. What does the phosphorylase do? Well, it goes around taking phosphate off of proteins. So when you do that, you activate one enzyme activity and you inhibit the other enzyme activity. And that results in more fructose 2,6-diphosphate, which does the opposite of what you have up here. so it decreases blood sugar, turns off gluconeogenesis, turns on glycolysis. So glycogenesis, making glycogen from glucose. So there was a problem, which was solved. The problem was simply Le Chatelier's principle. And so I remember the first time as a student looking at this, and I was like, well, wait a minute, what is this? What the heck is UDP for? I didn't understand its role. And yeah, it was never explained to me. I had to figure it out myself. But this is the reason why it's used. So it costs energy to make a glucose UDP, but at least you're able to circumvent this problem right here. Otherwise, you wouldn't be able to make glycogen. So does it cost energy to store glucose as glycogen? Yes, it does. Everything costs energy. So what about glycogenolysis, which is the release of glucose? Well, you have to break the the alpha 1,4 bonds. So let's see. And if you want to go past that, if you want to break down the branch points, then you need another enzyme.
Does glyconeogenesis take place in the mitochondria also?
No, that's outside. That's in the cytoplasm.
So the glucose 6 phosphate, the phosphate's removed and then it goes back out of the mitochondria and then this UDP is added?
No, it doesn't really involve the mitochondria.
Oh, okay.
Yeah, because glycogen is stored outside the mitochondria, right? So yeah, so there's control over glycogenolysis and glycogenesis. You can't have the two processes going on at the same time because that would be ridiculous. So phosphorylation of the enzyme activates this activity and inhibits the other activity. Okay. So these two separate enzymes right here have opposite effects when you put phosphate on them. One is activated, one is turned off. So here's kind of that process right here. So here's the pyruvate kinase. It's also a control point. It's activated by insulin and inhibited by glucagon. Okay. So let's see. And then, okay, here's the transport stuff. So how does pyruvate get into mitochondria where the citric acid cycle is going on? Well, you need a translocase. It's down here. Okay. So freely permeable with the outer membrane. Here's the inner membrane space, but the inner membrane is actually pretty impermeable to most stuff. Remember, it's about 70% protein. It contains also the electron transport system. So this inner membrane right here is just packed with protein and enzymes and all this stuff. So as pyruvate is brought in, it's also bringing in a hydrogen ion in a co-transport type of situation. So you're consuming some of the electrochemical gradient by bringing in pyruvate. Not very much, we don't really keep track of it, but it does take some. So that's how you get the pyruvate in there. What about ADP and ATP? Okay, well, notice that ATP goes out as ADP comes in. Okay, but it's freely permeable throughout the outer membrane. Okay. What about phosphate? Again, phosphate is brought in by symport with a hydrogen ion, so it costs energy to do that. So here's what the pyruvate dehydrogenase complex does. It puts acetyl coenzyme enzyme A on it over here after it decarboxylates this right here. At the same time, you're making an NADH, which is worth 3 ATPs. So this is in the mitochondrial matrix, so you instantly make 3 ATPs. So what about the NADH that was produced in the cytoplasm? It can't get in directly. And here's another, sorry, I have to keep watching my email. But here's another kind of important concept. So the NADH that's in the cytoplasm is kept separate from the NAD and NADH that's in the matrix. So you don't ever want to have a way to get, so you might end up with all of your NADH in either the matrix or in the cytoplasm, because then the systems will shut down. So those are two separate pools of redox agents that don't go back and forth. If that makes sense. So here's the matrix, excuse me, the cytoplasm out here. So NADH produced from glycolysis combines with oxaloacetate. And what are you doing? You're reducing oh, you see oxaloacetate again, don't you? And it's oxidized into malic acid. And then malic acid can come in with an antiport mechanism with alpha-ketoglutarate. Okay. So then what happens? The malate can be reoxidized into oxaloacetate. Okay, so oxaloacetate, during that process, you're making NADH, which simply goes and makes ATP. But now, to complete this cycle, oxaloacetate is converted into aspartate, and then aspartate can go out in an antiport with glutamate. So you've seen the role of glutamate in a couple of different things besides being neurotransmitter. You turn it into glutamic acid. Can you imagine what would happen to the cell if you simply shut down this transporter right here, or this one right here, or this enzyme right here? You would really be messed up, wouldn't you? So here's the other less efficient way to get NADH from the cytoplasm into the matrix. So here you're using dihydroxyacetone phosphate, and you're reducing it to glycerol 3-phosphate. Okay, well, the glycerol 3-phosphate can be brought in, and if you look at this is really kind of an antiport. You don't have to involve all these other different things over here. So you can see it's a more simple process. And you use FAD to oxidize it into dihydroxyacetone phosphate, which can then simply go right back out again. And FADH2 enters complex 2, and so it makes only 2 ATPs. So this is less efficient. That also means that it generates heat. So questions before we go into citric acid cycle? So all of this is in the mitochondrial matrix. Well, not that though, but and not that, but this part is, I should say. Oh, yeah. Okay. So here's where pyruvate is turned into acetyl coenzyme A. Okay. And so this red line right here is not separating matrix from cytoplasm. It's separating the citric acid cycle from essentially pyruvate dehydrogenase. So it comes in with acetyl coenzyme A. Now, some people talk about acetyl coenzyme A as being kind of a tweezers. So this is a pretty small molecule. So it would be pretty difficult to get that small little molecule and make it do all the things that you want to do. The enzymes have to have a way to hold on to it. So it's kind of like a molecular tweezers. So the first enzyme kicks out the acetyl coenzyme A as it combines it with, not acetyl, but coenzyme A, as it combines acetyl with oxaloacetate. So here's 4 carbons, you add 2 more, now you have 6 carbons. This is citric acid cycle, the name of the cycle. So then you turn citrate into isocitrate by a series of reactions that results in that enzyme really being an isomerase. And then you can oxidize isocitrate into oxalosuccinate. Well, we just made 3 ATPs. Then we decarboxylate and we get alpha ketoglutarate. And then the next step is very complicated enzyme. What it does is it decarboxylates again, so that's the 2 carbons you brought in are now gone. They're not the same exact carbons, by the way. And then at the same time, you're binding NAD, so you're making another NADH, another 3 ATPs. And then coenzyme A is brought back in again. Okay. So now we have succinyl-CoA. And notice that it's still a carboxylic acid. Let's see. And the next step occurs on the surface of succinyl-CoA synthetase, a substrate level phosphorylation. And so again, coenzyme A comes in, taken right off. I guess it's needed for this enzyme right here to be able to bind it. So then you're making substrate level phosphorylation, not using ADP, but using GDP. So the immediate product of this enzyme is GTP. And a subsequent enzyme turns GTP into ATP. So with all that, you end up with succinate. And then the succinate dehydrogenase is simply an oxidoreductase, but it uses FAD. You're oxidizing succinate into fumarate. So remember, we talked about fumarate as being one of the products of the urea cycle, and this is where it would enter. Okay? So remember, parts of the urea cycle use the matrix. So anyway, we made an FADH2, which is worth 2 more ATPs. And then, so we got fumarate. It's converted into malic acid. And then malic acid is oxidized into oxaloacetate. And again, you're making an NADH, which is worth 3 ATPs. So when we're talking about the urea cycle, and I was saying that fumarate is worth 3 ATPs. Here's the reason why. So here's one turn of the cycle right there. And this is simply adding up all the things that give us energy. In fact, that gives us 2 ATPs. This gives us 9 more, right? So we say that acetyl coenzyme A through the Krebs cycle is worth 12 ATP. So here's 9, 10, 11, and 12. One comes from substrate level phosphorylation. So I made a handout, or not handout, but a figure where I took the citric acid cycle and I just added all the control points. But somehow I thought it wasn't that important. Okay. In a way, it's kind of obvious. Look at this one, for example. Before this cycle, pyruvate carboxylase is inhibited by acetyl coenzyme A. Why would that be the case? Because that's the product. That's simple product inhibition right there. Pyruvate dehydrogenase is inhibited by ATP, NADH, and acetyl coenzyme A. Well, all 3 of these things would indicate that there's a lot of energy in the matrix. There's a lot of energy. Why do you need to make more? Same thing here. Same thing here. Same thing here. Okay. And this part right here, oh yeah, that would activate it, wouldn't it? So here's the overall reaction before the electron transport system or without oxygen. Here it is after electron transport or with oxygen. Here's the places, the different steps where we actually make that energy. So we've gone all the way from glucose to carbon dioxide. So now we can measure up how much energy we're making by adding up the ATPs and such. So if we're using this malate aspartate shuttle, we can make up to 38. If we're using the glycerol 3-phosphate shuttle, we can make 36. So then we can actually measure biological efficiency. So we saw a couple places where oxaloacetate was used, meaning that it's necessary for those processes. So there has to be ways that you will never run out of oxaloacetate. And as I listen to the words that are coming out of my mouth, I'm thinking, wow, what a great scam I could have here for a supplement. I can just sell people oxaloacetate. Anyway, so here's one way via phosphatidylpyruvate. Here's another way, and then here's another way. Okay. And we talked about the transhydrogenase, and I probably should be more careful about this. So now that I'm thinking about it, I should put, 2 purposes of the transhydrogenase. One is to generate NADPH, for the purpose of glutathione reductase, and the other is to regenerate NADPH for the purpose of biosynthesis. So in this class, we don't have time to go in very much biosynthesis, but here is one short pathway. So here's glucose 6-phosphate. It's been shunted away from glycolysis. So notice that you oxidize to make NADPH, right? Why would you do that? Because you need it for biosynthesis. Another enzymatic step, and then another place where you're making NADPH, and you end up with ribulose 5-phosphate. But that can be turned into ribose and deoxyribose for synthesizing nucleic acids. At the same time, the NADPHs are going to be used for biosynthesis of whatever else might be needed. Okay. Be right back. I need to get something to drink here. Okay. So the metabolism of lipids. So stored fat in adipose tissue can be hydrolyzed, mobilized in the blood. We know that by lipoproteins. And so lipase breaks it down into glycerol. That can enter glycolysis. And the 3 fatty acids will go through beta oxidation. So beta oxidation, I tried my best to simplify it. So we have 2 basic steps. One is simply activation. That means getting the fatty acid into the mitochondrial matrix where beta oxidation occurs. And so activation means you have to make it into a coenzyme A. So you have to put the tweezers on it to get it in there. So the tweezers are necessary. And then it's able to interact with carnitine. So there's an enzyme that takes the activated fatty acid, combines it with carnitine instead of coenzyme A. But if you don't have coenzyme A on it, then this enzyme can't work on it. So now this molecule can then come through the transporter. But the carnitine is then removed, so the pool of coenzyme A is used to knock off the carnitine, and the carnitine can just go back over here again. What's a transporting system? Without carnitine, you don't have this process. So here's carnitine. It's an amino acid, although this is a quaternary ammonium. I guess that's necessary to get it through the transport protein. Okay. And here's how you form the ester linkage with the fatty acid that you're going to oxidize in the carnitine. So then once that's in there as acetyl, or the acyl-CoA, acyl means this part if you remember, right? Then you go through degradation. So maybe I'll just look at it this way. So here's the cytosol, here's the mitochondria, okay? And so they're showing you the part where they're putting, so here's the fatty acid, so here's coenzyme A. They're not showing you the part where you need carnitine to bring it in. nothing's perfect, you know. I steal this stuff, and then the stuff you steal is not even perfect. It's so discouraging. So notice the first step is it's oxidative, right? You're just oxidating this fatty acid. So here's the carbonyl carbon, here's alpha, here's beta. So everything happens concerning the beta carbon. So the first thing you do is you make a beta alpha double bond, and you made an FADH2, which is worth 2 ATPs. And then you add water to that. Now you have a beta hydroxy. And then you use NAD to oxidize that secondary alcohol into a ketone. And then see where that yellow arrow is, because here's the 2 carbons that you're going to cleave off using coenzyme A. So it comes in and just replaces this bond right here. And this is the acetyl coenzyme A that goes off and then goes into the citric acid cycle. And then you keep going until you run out of carbons. So if you have an unsaturated bond, it has to be isomerized into trans and then hydrogenated. You have an odd number of carbons. The last oxidation will leave propanoyl coenzyme A instead of acetyl coenzyme A. That alpha carbon is oxidized to a carbonyl and then removed to CO2, leaving acetyl coenzyme A that then goes into the Krebs cycle. If you look at the energy expenditure, activation needs 2 ATPs. Then every turn of the cycle, you make an FADH2 and NADH, and you make an acetyl coenzyme A. So every cut gives you this, and at the end, you end up with 1 extra. That's the one last acetyl CoA that's left over. So looking at this fatty acid, I would have to cut 1, 2, 3, 4, 5, 6, 7, 8 times. So I made 8 cuts. So I did this 8 times. So I got 8 FADH2, 8 NADHs. And then when I cut it 8 times, I have 9 pieces. Each piece is acetyl CoA that's worth 12 ATPs. So we went into lipogenesis a little bit, and this particular enzyme right here, acetyl-CoA carboxylase, we talked about in terms of dieting and stuff like that, because of the effect of exercise on it. So anyway, in this case, we need what's called an acyl-carrying protein. So don't forget, this occurs in the cytoplasm, right? Whereas this is in the matrix. Okay. So you have the acetyl coenzyme A, and again, there's the tweezers. So that tweezers is replaced by better tweezers, the acyl carrying protein, the one that's modified for the enzyme ACC. It takes ATP to do that. And then, this is 2 carbons. It takes a CO2 molecule from the cytoplasm, and at the expense of another ATP, puts it onto acetyl-CoA and turns it into malonyl-CoA, which is 3 carbons. And then it takes that acyl carrying protein that you spent money on up here, and then whatever you made right here from ACC, and you're using 2 reducing equivalents. Again, this is biosynthesis, so NADPH. And you add the 2 carbons to each of the 2 carbons, and you get 4 carbons, and one of the CO2 is given off. So by releasing that CO2 and spending all this energy right here, you ensure that this reaction is a one-directional arrow. And so you simply repeat, repeat, repeat until you end up getting all the carbons that you want. And finally, an enzyme hydrolyzes the fatty acid CoA from the acyl-carrying protein to release the fatty acid as the fatty acid. So when I did this calculation here, I started at the level of acetyl coenzyme A. So how much ATP would be required to synthesize an 18 carbon fatty acid starting with acetyl coenzyme A? Well, the first step with the acyl carrying protein is going to require an ATP. Each of the subsequent steps is going to require 8 ATPs. And then the reduction requires another 48, so the answer was 57 versus 38 that you would get from oxidizing it. So it's another example to look at biological efficiency. In this case, it's about 67%. Well, then how do you make phospholipids and cholesterol and things like that? Well, again, biosynthesis takes energy. So glycerol is phosphorylated. That takes energy to do that. And then you have synthetase enzymes that put a coenzyme form of the fatty acids onto the alcohols. So this thioester is converted into a regular ester. And so here's a few other pathways. So you can see glycerol turning into glycerol 3-phosphate. And then here's dihydroxyacetone phosphate. You take this and you combine it to make lysophosphatidic acid. That means it's got 2 fatty acids on it, not 3. And then you add another one. Let's see. And then, yeah, so here's a diacylglycerol. Add another one. Now you have a triacylglycerol. And sometimes they need inositol to make those kinds of. But this is an example. Skipping a lot of steps. So cholesterol can be synthesized in the liver from acetyl coenzyme A. And here's where the statins are involved to stop that process. Here's the 2 enzymes that are mostly inhibited by the statins. So here's where we're talking about regulation and that enzyme ACC. So you have several isozymes of ACC. Some are more responsive to allosteric effectors than others are. But ACC2 is inhibited by exercise. And they found that mice whose gene was knocked out could not get obese. So if we look at what the enzyme responds to, it's turned on by insulin, and glucose is turned off by glucagon and by exercise. So exercise makes it so you don't have a lot of melanoCoA. So that activates fatty acid oxidation, inhibits fatty acid synthesis, and also activates carnitine palmitoyl transferase, which does what? Brings the fatty acid into the mitochondria for oxidation. Okay. So, if we found an enzyme inhibitor for this, we can make everybody skinny and make ourselves rich. So my point with this, I hope, is kind of clear. Yeah. How do you cause insulin to be released? Okay, for the most part, it's high sugar in the blood. High sugar in the blood causes all kinds of problems with glycation and all that kind of stuff like we talked about with diabetes. So releasing insulin in the presence of high blood sugar is a way to protect your body. So insulin not only puts glucose back into cells, it also puts triglycerides back into cells. So that's the reason why if you want to burn fat, it's not good to cause an insulin spike. Don't drink sugary stuff. Yeah. So any questions on that chapter, which was kind of long? A lot of stuff in it. Like you think you don't have to memorize it, huh? When I was a student, they just had everybody memorize everything. I remember taking Biochemistry 441, and there was a blank page, and they said, write out the Krebs cycle. Yeah. Oh, I see there's something in the chat. Where does UTP come from in glycogen synthesis? Well, it's just all throughout the cell. I mean, UTP is like ATP or, GTP. It's just uracil instead of adenine, right? So it's all over the cell. And it says I'm sharing, but I don't see the green box. There's the green box. So I'm still working on the organization of these later chapters. When I decided to write my own book, I decided that I'm going to just close my eyes and try to think about it. What is the best way to do this, and not just copy what the other textbooks do. So diabetes is a multivariant disease. It's got so many factors. There's a genetic component. There's a strong environmental component. All kinds of different things. So diabetes is a risk, especially with now this COVID-19. It's a risk factor. It's a risk factor for almost everything. And how all these risk factors kind of add up really is in the form of inflammation, like we talked about in another chapter. So I think we know what pancreatic alpha and beta cells do. So type 1 is insulin dependent. Type 2 is more insulin resistant. That means that the cells don't have the receptors necessary to respond to the insulin. And the problem with high glucose is glycation. Okay, so this is known to damage beta pancreatic cells responsible for producing the insulin. So it's a vicious cycle. And then what happens when beta pancreatic cells are damaged? They release cytokines, which are messenger proteins and other molecules that attract the immune system. So now if you get the immune system involved, they may, B and T cells may actually be attacking beta pancreatic cells. So now it has an immune component to that disease. So here's the ketosis part. So here's acetyl coenzyme A. The carboxylic acid isn't there because it's an ethioester. So there's nothing acidic on this. When you hydrolyze it off, you might get acetoacetate, which is an acid. So most carboxylic acids at physiological pH are going to be ionized like this. So where this one had no acidic hydrogen, this does. It produces hydrogen. So that's acidosis. So the more ketone bodies that you make, the more acidosis you have. So then acetoacetate can be hydrolyzed into acetone. Okay, so that also gives the proton over there. It can be reduced into beta hydroxybutyrate, but that's also an acid. Energy depletion, what's going to be caused by severe acidosis and stuff, causes more acidity to build up. So here's some genetic diseases, and I just tried to give you a list of some that just happened to pop in when I was thinking about things. And a lot of this came from my friend who passed away a couple of years ago because I helped him edit his last book. It was on medical biochemistry. And he put a lot of case studies in there, and that was really, really interesting. So here's autosomal dominant, here's autosomal recessive. Dominant means that if one of the parent has it, half of the kids are going to get it. So anyway, recessive means that you need 2 copies of the gene. So if both are carriers, right, this is the pattern that you would get. About 1 out of your 4 children are going to have the disease, even though you don't have the disease. Here, this person has the disease, and half of the offspring will get that disease. So then another category of disease, which really means what it is, right? Disease. It's not ease, it's dis-ease. Accumulation of various metabolites. So lactic acid is something that we experience all the time. Gout and kidney stones, we can avoid. We can avoid this as well. But nevertheless, they're disease, aren't they? And then you get to some that are very severe. Like PKU is a pretty severe disease, especially if you're a baby And nobody knows that you're not supposed to have phenylalanine. And the lysosomal storage diseases, they're fatal. Accumulation of very long chain fatty acids, fatal. Cystic fibrosis is fatal. Okay. And so, look at this. It's an autosomal recessive disorder caused by a mutation in both copies of the gene for the membrane transport protein. And when that's not functional, secretions get thick, hard time breathing. Is very similar to what people are dying from COVID-19 is. They lose the ability to exchange respiratory gases. So this can build up. Homocysteine can build up. And this is actually an indicator of risk for heart disease. BMAA is another example of an environmental toxin, but it causes a disease, doesn't it? Then we talked about autoimmune diseases. They may not be exactly metabolic diseases, but most diseases have several different, what's the word for it? Causes or multiplying effects. So antiphospholipid syndrome. So you think, oh, it's simply an autoimmune disease, but okay, yes it is. But why would their antibodies attack, say, prothrombin? Something has gone wrong in that system. And yes, so you look at a disease like this and you try to decide, well, is it genetic? Do people inherit this disease? And it gives you a little bit of information about how you might approach a cure to that. So we talked about lupus. My one friend with lupus, I do see her post stuff on Instagram and stuff, but haven't seen her for a while. She's hiding because she knows that if she got the COVID virus, it would be detrimental to her. So celiac disease is an immune response, isn't it? But then why is some people able to handle gluten and other people not? So that becomes a genetic issue, doesn't it? I was going to put Crohn's disease and never got to it. And yes, diabetes could also be an autoimmune disease. And could it be also caused by a virus? Yeah. Something went wrong genetically. We know what viruses do now. That's a possibility. Let's see. Then we talked about brain diseases. So Alzheimer's, we spend a lot of time on that, especially about the drugs, because we don't have a cure for Alzheimer's. I'm not sure we have anything that can even slow down the progression of Alzheimer's. Then when you look at people with Alzheimer's, some of it is what we call early onset. They get it way before they're supposed to. Sometimes people are diagnosed and then the disease progresses extremely rapidly. But most people, it's not very rapid at all. It's just a slow decline. Okay. So immunity in the brain, this is really a brand new thing. This is in the last 5-10 years. And, really interesting stuff going on there. And then I talked about inflammation, which is really the thing that, how do you say, makes all the different processes in your body accumulate, I should say. Because every little malady is going to increase the amount of inflammation. It's really inflammation that is the best indicator of what you're going to have a heart attack and stuff like that. So I put in a little bit about, well, bacterial and viral infections. And we already talked about viruses. There was a whole chapter on that. Didn't really talk too much about bacteria, but we know about these diseases. So tuberculosis is in my family, actually. So when I was about 8, I think, my dad was diagnosed with tuberculosis. He apparently contracted it during World War II, and it simply devastated our family. I mean, he ended up in Leahi Hospital for 5 years. We never knew whether he was even going to come out alive or not. But yeah, it's devastating. And that wasn't even drug-resistant form. So there's bacterial collections, and people can use that to study bacterial evolution, to test all kinds of different methods and detection methods and everything else. And gene sequencing of known pathogens can be used to figure out what's going to happen. So this whole field of bioinformatics is becoming amazing. And it's all possible because of the computer capabilities. So any questions on anything in this chapter? Okay. Oops. Can't share until I open up the file. So this whole chapter is a long chapter, but I think it's maybe one of the most important chapters because you study all this stuff. What is the end point? What do you get out of it? The purpose should be that all this information helps us be able to lead our lives in a better way. So this is really a good culminating chapter. Although it's more or less for your edification, it's not really something that is going to supply a lot of questions on an exam. So yeah, nutrition, I went into a little bit of nutrition, but it's not meant to circumvent a nutrition course. If you take a nutrition course, there's a lot of stuff that you won't learn about that we talk about in here. So I went into a historical and functional explanation of enzyme cofactors and enzyme substrates, of which many are vitamins and minerals that are essential. So it's kind of funny, in some parts of the chapters in disease, we're talking about metabolites that, or there's too much. But in this one, there's not enough. Typo. So let's see. So this whole thing on essential fatty acids really should tell you that you cannot go on a fat-free diet. You will suffer from it because there's essential fatty acids that you need. Let's see. Here's the cascade of all these signaling molecules. If you don't have enough arachidonic acid, for example, you're not going to be able to have all these signaling functions. Your body won't function correctly. And then I did attack the industry of supplements. These people are making millions of dollars lying to you. And I'm sorry, I have a little bit of, morality about it. I don't like it when people lie and they get away with it and make money. But your nutrition advice has to be based upon experiment, on evidence. And people are trying really, really hard to get good data. Then that data is just circumvented by people that are selling you stuff. So just to show you how things can be wrong, it's like, look at the deal with salt and blood pressure. Most of the medical field to this day is wrong about it. Matter of fact, I do have a meeting scheduled with my department chair, who's an MD, PhD, and he specializes in cardiology. I should ask him that question, just to see what he says. Now I'm looking forward to the meeting. Let's see. Iron, I think, is one of the major deficiencies. This whole thing about red meat, white meat, plant-based, all this kind of stuff, it's just like, okay, that's correlated to all kinds of other things, but in the end, you still have to get iron somehow. So dieting, because obesity is going way up, and obesity is correlated to increases in all kinds of diseases. Let's see. So in the end, what are we really trying to do? We're trying to get enough of the essential nutrients to keep our body in good shape. When people go off and do weird diets, the keto diet is a weird diet. Being a vegetarian is a weird diet because our DNA, our digestive tracts, are not that of a vegetarian. They're that of an omnivore. So what is a good quality food? That's a food that has a lot of essential nutrients relative to the number of calories that's in that food. So when I go buy a prepared food, which is like, how do you say, what's the word for it now? Highly processed, they're going to put fat in it because that tastes good. That means you're going to go back and buy that product again. So that may not be a high quality food because to get that amount of nutrients, you have to take in a lot of calories that aren't really quite necessary. But yeah, good luck trying to find any data on that. If you type in high quality food, you're going to come up with stuff like, books and supplements and Whole Foods and all this kind of stuff, right? Whole Foods, the company. There's actually a term now Orthorexia nervosa refers to a condition when a person is obsessed with eating only foods that they think is healthy. So can food be toxic? Yes, it can. So I tried to give you several examples of that. And you know what? I'm not here trying to defend processed food and talk smack about organic foods. All I'm saying is that you got to get your essential nutrients no matter what that food is called or how it's being sold. You need to get your essential nutrients. So cooking can cause problems. But not cooking also causes problems. And then food allergies, we probably have the wrong approach to food allergies. Right now, food allergies are like, oh, that's food's bad, you got to stay away from it. That's almost ensuring that your kid is going to develop allergies if you protect them from everything. Kids should be outside playing in the dirt and doing all this kind of stuff to build up their immune system. So we're out of time, but the microbiome, I put a lot of words about that in there. And then exercise. I really tried to emphasize the fact that exercise is medicine. Food is not. Food, you just get your essential nutrients and that's it. But exercise, especially for the human species, is extremely important. For the monkeys and apes, nowhere near as important. But human metabolism is this hyped out. We're supposed to be running down food, what we're supposed to be doing. You know, not driving to Starbucks and getting a non-GMO muffin. We're supposed to chase down, hunt, and kill that muffin on our own. So then I tried to put some stuff that's probably good for a healthy lifestyle, as best as I could come up with what's out there. So these are two good friends of mine. So there's a lot of interesting stuff that they put in this one article from a couple of years ago, and I think it's worth reading. So they have both gone through the whole gamut of health fads. They've been, athletes. They've been vegetarians. They've been all kinds of stuff, and now they're omnivores. Pretty interesting. So does anybody have any more questions about anything?
I'm just curious for this test, is it going to be like essay questions or just like 1 or 2 sentence answers?
Well, I won't put essay questions. There'll be short answers, if anything. But there'll be a lot of true-false questions referring to metabolic pathways. Maybe some other questions about metabolic pathways, but yeah, I don't give essay questions. At the most, just short answers.
Okay, thank you. And then also I was trying to look at the figure from chapter 39, and I think it's not loading on Laulima. I don't know if it was just me or if anyone else had that issue, but it said the PDF is not viewable.
Oh,
I couldn't see it either.
The figure one?
The figure from chapter 39.
Okay, 39. Oh, table one.
Yeah, sorry, table.
Okay. You know what that is? Oh, I see. That's not even opening here. Hold on. What that is all the the structures of the
I think you muted yourself, Professor.
I think you're on mute, Professor.
So what, you guys learned how to mute me after all this time?
We totally missed everything you were saying on the table.
Oh, okay. So yeah, I will upload a new PDF version just to get it up there for you. But all it is really is just the structures. Okay. Can't tell if it's being shared or not. I must have hit a different button. So this is what it is. It's just the structures. Yeah.
And then one more question. For the extra credit, is there like a specific rubric we should follow, or is it just like our research into something?
It's just free form. Do whatever you feel like doing. You know, it's I'm really looking at how did you respond? How did it change you to take this class and to learn this information? So, and I think you should be interested enough in a disease or some aspect of nutrition or something to write a page about what you think is cool about it. You know, it's kind of the idea. So I don't have a specific rubric.
Okay, sounds good. Thank you.
What other classes do you teach? Do you teach OCHEM?
Not anymore. I used to teach that for several years. I don't teach it anymore. I found somebody who does a good job with it, so I just let her go with it. And that happened when I was faculty senate chair. I was faculty senate chair for 4 years in a row, so I had to give up a course, and that was the course I gave up. Yeah. Yeah. Okay, well, email if you have any other further questions. I'm going to be in this office all day and into the evening tonight. So you guys have a great day and a great weekend, okay?
You too.
Thank you. Take care.
Thank you.